This is a precision-engineered dry floating platform. It is designed to drive targeted acoustic patterns through a human subject. But the delivery of these waves presents a biophysical paradox. We have to identify the exact mechanism by which a macroscopic sound wave triggers specific microcellular biological changes. Qualitative descriptions like vibrational healing are insufficient for clinical or bioengineering standards. They lack a defined, measurable pathway. To solve this, we must look to sonobiology and classical mechanotransduction, the study of how cells sense and respond to physical forces. This process can be tracked as a single continuous chain of events, moving from the device through the density of the skeletal system and arriving at a specific receptor protein docked on the cell membrane. The ENTL system utilizes acoustic impedance matching to prevent energy loss, converting mechanical pressure into biochemical signals via the focal adhesion kinase, or FAK, pathway. Validating this technology requires treating it as a strict engineering problem, tracking the direct conversion of external mechanical energy into localized cellular intelligence. Airborne acoustic therapies are physically limited because air heavily attenuates mechanical energy. The pressure waves lose significant power before they can reach deep tissue. The platform overcomes this through acoustic impedance matching. It uses a water-based transmission layer that couples directly to the human body, which is itself approximately 70% water. Because the densities are aligned, the waves enter the body through interstitial fluids without the energy decay seen in air-based systems. Once inside, the skeletal system functions as a high-speed network for mechanical energy. Dense bone transmits mechanical waves at 3,000 to 4,000 meters per second. This speed and rigidity allow the frequencies to bypass the damping effects of soft tissue. This utilizes the same physical principle as bone conduction, a natural pathway human hearing uses to transmit sound through the skull. Mechanical frequencies travel through the dense temporal bone to reach the cochlea and vestibular organs directly, bypassing the auditory canal. Systemic acoustic therapy relies on this capacity of the skeleton to act as a lossless conduit for precision frequencies. To understand the cellular response, we must look past the bone lattice to the extracellular matrix surrounding a single cell. The cell is physically linked to this matrix through connection points called focal adhesions. When an acoustic pressure wave travels through the skeleton, it places direct mechanical stress on these anchors. Research from Kumeta and colleagues in 2025 demonstrated that audible range acoustic pressure triggers specific biochemical changes at these exact sites. This diagram shows the mechanosensitive protein responsible for this transduction, focal adhesion kinase, or FAK. Acoustic stimulation causes FAK to undergo phosphorylation at tyrosine 397. This phosphorylation is the critical event that converts a raw physical force into an actionable biochemical signal inside the cell. The FAK pathway provides the biophysical link, proving that cells possess the hardware required to translate acoustic pressure into biological instructions. This activation initiates a downstream signaling cascade that reaches the cell nucleus. The first genetic response is the upregulation of the mechanosensitive gene PTGS2. This leads to the increased synthesis of prostaglandin E2, a key signaling molecule. These biochemical signals force the cell to remodel its internal scaffolding, or cytoskeleton. The resulting physical changes include enhanced cell adhesion and the formation of lamellopodia, structural extensions that allow the cell to move and stabilize. In the 2025 study, this acoustic stimulation effectively suppressed the differentiation of fat cells. This means a targeted sound wave physically prevented a cell from turning into a specific tissue type. Acoustic mechanotransduction allows for the active mechanical regulation of cellular expression and architecture. This mechanical energy travels deeper than the cell membrane, entering the microtubule network. Microtubules are hollow protein tubes that form the cell's internal structure. Research indicates that microtubules generate coherent electromagnetic oscillations as they vibrate. This resonance is sustained by structured or exclusion zone water that lines the microtubule cavities. Near cell membranes, water forms highly ordered, hexagonally structured layers known as EZ water. This layer separates negative charge and reduces thermal fluctuations, which protects the subcellular vibrations from noise. The synergy between microtubule oscillations and this structured water allows the cell to maintain vibrational coherence. Precision acoustic vibrations from the platform create the hydrodynamic shear required to stabilize these structured water layers. Maintaining this subcellular coherence supports the cell's ability to process information and maintain biological order. Scaling back to the macro view of the entire human body, we can now see how these individual reactions integrate. Trillions of simultaneous vac transductions and microtubule vibrations combine to drive systemic physiological changes. These shifts are registered by the heart, which generates the body's most powerful electromagnetic field. The whole body resonance of the platform aligns these cellular oscillations with the heart's organizing toroidal field. This engineers the resonant environment required for the body's innate self-regulating mechanisms to function optimally. By bridging cellular physics with systemic resonance, acoustic dry floating provides a measurable pathway for restoring biological coherence.